Okay, in this lecture we talked about the nervous system. We mostly talked about basic physiology of the nervous system and the components of the nervous system, but we also talked about a couple of diseases. There will be two videos. I will do one here covering most of the introductory stuff, and then the last one will cover whatever I don't cover in this video. So the nervous system has two main components, so it's broken down into the central nervous system and the peripheral nervous system. Now the central nervous system includes the brain and the spinal cord, and the peripheral nervous system is composed mostly of the somatic nervous system and the autonomic nervous system. There's also this little, the enteric nervous system, which is really a component of the autonomic nervous system, but some kind of throw it in here as a part of the peripheral nervous system. Now, the autonomic nervous system is further broken down into the sympathetic and parasympathetic divisions, and I'll talk more about those in a little bit. Now, this note that the cranial nerves are components of the peripheral nervous system. This is important because a lot of people think, or when they hear cranial nerves, they think it's the central nervous system, but it's not. They are peripheral nerves. There are actually a couple of exceptions, but we're not going to worry about that. They are considered components of the peripheral nervous system. So the nervous system as a whole, its job is to sense, to integrate, and then respond to different stimuli. And there can be any type of stimulus. There's all sorts of stimuli. Nervous system has neurons. These are the conductive cells. These are the cells that are going to conduct action potentials in the nervous system. Plus the neuroglia. Neuroglia means nerve glue, and these are supporting cells of the nervous system. So the main components of a neuron, a nerve cell, is the cell body, which is also known as the soma. Sometimes it's called the perikaryon, the dendrites, and the axon. There's only one axon per cell. Most neurons will have many dendrites, with a couple of exceptions, but all neurons have one axon. So these neurons can be classified as sensory, motor, or interneurons. The sensory neurons are also known as afferent neurons. The motor neurons are efferent, so these are the ones that are sending information out. The sensory ones are receiving information. And then interneurons, they're basically connections between the afferent and efferent neurons. And neurons communicate with each other by neurotransmitters, and I'll talk about some of the important neurotransmitters a little bit later. So here's a typical neuron. So this is the cell body, also known as the soma or the perikaryon. This is the axon. This particular axon is myelinated, which I'll talk about in a little bit. This is important for increasing conduction speed down the axon. These are the dendrites. These are important for basically bringing information into the neuron. Information will then leave through down the axon, and these are synaptic terminals. Again, I'll talk about that when we talk about synaptic transmission. So some of these neuroglial cells, starting with the neuroglia of the central nervous system, remember these are the supporting cells. These are the nerve glue. And we're going to talk about the neuroglia of the central nervous system first. So astrocytes, these are the most numerous of the glial cells. They're very large, and they're really important for a number of functions. So they form part of the blood-brain barrier. They regulate ion flow within cells. They're important for signaling. They have a lot of jobs, and honestly, they do probably more than's understood. Oligodendrocytes, these are cells that produce the myelin sheath. They myelinate axons in the central nervous system. They're also important for regulation of pH in the central nervous system. Microglia, these are cells that basically come from monocytes. That's what I mean by the mononuclear phagocyte system. And basically they're phagocytes. They're immune cells that are in the central nervous system. And this is actually pretty unique because it was for a long time believed that the brain and the central nervous system was pretty much free from immune cells, but these microglia really are immune cells. Ependymal cells, these basically line the ventricles of the brain and the brainstem, as well as the central canal, and they produce cerebral spinal fluid. So up here we have astrocytes. Again, they're very, very large. There are many of them in the central nervous system. Down here we have these oligodendrocytes. They're kind of unique because they can myelinate multiple axons at a time, which is pretty cool. The microglial cell, again, this is a phagocyte. This is an immune cell that is in the central nervous system. And then we have ependymal cells, and they are important for producing the transcellular fluid known as cerebrospinal fluid. Okay, so as far as the peripheral nervous system is concerned, there are far fewer neuroglial cells. The Schwann cells, these are the cells that myelinate axons in the peripheral nervous system, and basically what they do is they kind of wrap themselves around the axon, and then they just kind of do like this jelly roll thing where they continue to kind of wrap around, and that's really what forms the myelin sheath in the peripheral nervous system. Remember, in the central nervous system, it's oligodendrocytes that produce the myelin sheath. And secondly here, the satellite cells, these are really important for structural support in the peripheral nervous system. That's kind of their primary job. They probably do more, but again, they're not very well understood. Okay, so we talked about neurons, kind of reviewed neurons, and then we started to talk about action potentials. And these are basically the mechanisms, part of the mechanism by which neurons communicate with each other. Okay, they have to send information down the axon, and action potentials are the form that information takes. So action potentials, they are all or none events. They either occur or they don't. You don't just have like a little action potential. They either occur or they don't, all or none. And this occurs basically in response to a stimulus, okay? And that could be anything. I mean, any sort of stimulus could stimulate an action potential. So occur in response to altering the electrical charge across the membrane by changing permeability to different ions. That's the key here. In order to communicate information down an axon, we have to change the permeability of certain ions, namely sodium and potassium. So the resting membrane potential, this is the membrane potential at rest of the nerve cell membrane. All right, and it's about negative 70 millivolts. So basically, the inside of the cell is more negative than the outside. Okay, and this is due mostly to proteins, because proteins are negatively charged. So these proteins that are inside the cell, this is what gives this negative 70 millivolt resting potential. And as we know, at rest, sodium is in high concentration outside of the cell, potassium is in high concentration inside the cell. And this is really important because since sodium is in high concentration outside the cell, it wants to come inside the cell. Same thing for potassium being high in concentration inside the cell, it wants to move outside. So that can be manipulated in order to move information down the axon. So if we alter the permeability of these ions, sodium and potassium, we can change the membrane potential. And again, like I said, this is how we are going to move information down the axon. And this occurs by opening channels. Opening sodium channels, opening potassium channels, because if you open a channel, sodium is going to come in. If you open a potassium channel, potassium is going to leave. All right? And this is going to occur in response to a stimulus, which is just a detectable change in the internal or external environment that can alter the resting membrane potential. And again, this could be anything you can imagine, mechanical pressure, heat, stretch, the list goes on and on. Now, if before we have an action potential, we have to reach threshold. All right. So basically, if the stimulus is powerful enough and we can change that membrane potential to about negative 55 millivolts, so it's becoming more positive, then an action potential will occur. If the stimulus is small enough and we don't reach threshold, there will be no action potential. All right. So that's what threshold is. That's the value that you need to get to before the action potential can actually occur. So let's talk about how this actually happens. So we reach threshold. So we've gone from negative 70 millivolts to about negative 55 millivolts. And as soon as we reach threshold, both sodium and potassium gates open. Okay. However, sodium gates open faster than the potassium gates. Okay, so if I kind of draw this down here. So this is going to be our nerve cell. Okay, again, sodium is in high concentration out here. Potassium's in high concentration in here. We have some stimulus, whatever that is. It could be anything. We've reached threshold, and now we are starting to go through the cycle of an action potential. So our stimulus is strong enough. We reach threshold. These sodium gates open. Okay, and potassium gates open also. So of course sodium is going to come in and potassium is going to leave. However, sodium comes in more quickly. Okay, so it comes in more quickly than potassium leaves. Think of it like a door. Okay, the sodium door is completely open, but the potassium door is only about halfway open. So we have more sodium coming in than potassium leaving. All right, and what happens is the membrane potential becomes more positive because we have more positive charge coming into the cell than positive charge leaving. So it becomes more positive, and this is called depolarization. Okay, so we're getting less and less and less polar. We're getting less negative. We're becoming more positive. And again, that's because sodium is coming in more quickly than potassium is leaving. All right, so this is going to continue until you reach what's known as overshoot, and this is when the membrane potential actually becomes positive by about positive 30 millivolts. Now at about this point, the sodium gates close. Okay, so the sodium doors close. So that means sodium is not going to come into the cell anymore. However, at this point, the potassium doors are completely open. So at this point, sodium is not coming in, but potassium is leaving as quickly as it can. And what's happening is you're losing positive charge from the cell. And when you're losing positive charge, the cell membrane becomes negative again. The membrane potential becomes negative. This is repolarization. So depolarization, this is when we were becoming more positive. Repolarization, this is when we're becoming more negative again. And again, this is just occurring very, very quickly down the cell membrane. Okay, these events. And that's how we propagate information down the axon. So potassium still continues to leave the cell, and what happens is membrane potential actually becomes more negative than that negative 70 millivolts value. And this is known as after hyperpolarization, sometimes just called hyperpolarization. Now, after hyperpolarization, this wasn't in your textbook, but this is also known as the relative refractory period, and that just means since we are even more negative than threshold, or more negative than resting potential, we have a farther gap to get to threshold. And that means it's just harder to have another action potential. All right. So that's what's meant by the relative refractory period. Now, there is a period known as the absolute refractory period, and this is when the membrane potential is above threshold. So when membrane potential is above threshold, as was occurring during depolarization, you can't have a second action potential. That's absolute refractory period. A second action potential cannot occur. Now, after these events have occurred, the action potential has propagated itself down the neuron axon. The sodium-potassium pump are going to kind of reset the concentration. So reset that high concentration of sodium outside and that high concentration of potassium inside. So this is the textbook image, and as you can see, so if we look at this top, so they have the resting potential at negative 80. That's actually probably more appropriate for most neurons. Muscle cells are about negative 70. I'm not too concerned about the numbers, but they have negative 80. So again, we have some stimulus. Sodium high concentration outside, potassium high concentration inside. There is some stimulus. And what happens is sodium will enter. Okay, we reach threshold. Sodium enters the cell. Potassium leaves very slowly. Sodium comes in much more quickly. So what happens is we become more positive, and this is depolarization. Then we have the overshoot where we become more positive. Sodium channels close. Okay, so sodium is stuck, and then potassium is leaving as fast as it possibly can. So you have positive charge leaving. The membrane potential becomes more negative. This is repolarization. And as this occurs, all the way down the axon, that's how we transmit information. So this is occurring all the way down the axon. Again, these action potentials are all or none events. Okay, so we talked about the action potential, basically how we move information down a single cell. Now let's talk about how one neuron communicates with another neuron, or even communicates with, say, a muscle cell. It's very similar. And this is synaptic transmission. And basically you have a two-cell setup. You have a presynaptic neuron. So if we look at this image down here, this is our presynaptic neuron. Okay, so this is the presynaptic neuron. And then we have a postsynaptic neuron. All right. And the space between these two neurons, between the presynaptic neuron and the postsynaptic neuron, is called the synapse, or the synaptic cleft. So that's in here. This is the synaptic cleft. So basically what's going to happen is you're going to have this action potential. It's going to work its way down this membrane, okay, just like we saw on the last few slides. It works its way down this axon membrane, and when it gets almost to this point, this portion of the presynaptic neuron is called the synaptic end bulb, and when it gets right to about this point, you're going to start having the opening of voltage-gated calcium channels. And what happens is calcium is going to enter this presynaptic neuron. Now, in this synaptic end bulb of the presynaptic neuron, we have synaptic vesicles. They're just membrane-bound vesicles that contain neurotransmitters. Neurotransmitters are going to be very important. So we have this action potential come down the presynaptic neuron. When it gets down to about this point, close to the synaptic end bulb, calcium enters. And what calcium is going to do is it's going to trigger the release of these neurotransmitters into the synapse, into the synaptic cleft. Okay? So these synaptic vesicles are going to travel down here. They're going to release their neurotransmitters into the synapse. And what those neurotransmitters will do, as we can see over here, is they're going to bind to receptors. Okay, so this is kind of like a blown up version of this. They're going to bind to receptors on the postsynaptic cell membrane. And when they bind, what happens is they open up. These receptors actually open up, and that allows positive ions like sodium and potassium to move into the postsynaptic cell. And what that does is that's the stimulus that the postsynaptic cell needs to transmit an action potential down it. So that's kind of the beautiful system of how these neurons communicate with each other. It's a really nice system. So just another quick review. Action potential comes down the axon membrane of the presynaptic cell. When it gets to the synaptic end bulb, that signals calcium to enter the cell. This signals the release of neurotransmitters into the synapse. The neurotransmitter binds to receptors on the postsynaptic cell. That stimulates these receptors to kind of open up and form a pore. This allows positively charged ions to enter the postsynaptic cell. And that's the stimulus that the postsynaptic cell needs in order to propagate that action potential down it. Now, a couple of things can happen after the neurotransmitter binds to these receptors. The neurotransmitter might be broken down in the synapse by enzymes. So, for example, a neurotransmitter like acetylcholine is broken down by acetylcholinesterase. It can also bind back to this presynaptic membrane and stimulate what's known as reuptake. So some neurotransmitters can be taken back up into the presynaptic cell, and some of the neurotransmitter will actually kind of just diffuse away. Okay, so this is basically the words of everything I just described. Like I said, neurotransmitter that doesn't bind to receptors is mostly going to be broken down by enzymes, or sometimes it is brought back into the presynaptic cell. That's known as reuptake. So again, this is just another picture. I think this is the one from your textbook, maybe. But basically, you have a nerve impulse, again, signals calcium to enter the cell, which triggers the release of neurotransmitter into the synapse, binds to receptors on the postsynaptic cell. That opens this pore in the receptor. Positive ions like sodium and potassium can enter, and that is the stimulus that this cell needs to propagate the action potential. Okay, then I talked about some of the important neurotransmitters, starting off with acetylcholine. This is a very well-studied neurotransmitter. We see it in the peripheral nervous system, central nervous system. So this is the neurotransmitter by which neurons communicate with muscle cells. It's important in the autonomic nervous system, so very important neurotransmitter. Glutamate, this is an excitatory neurotransmitter, so it stimulates other neurons, and this is seen chiefly in the central nervous system. So glutamate is the most common excitatory neurotransmitter in the central nervous system. GABA, this stands for gamma aminobutyric acid. This is a inhibitory neurotransmitter that's seen in the brain. So note the difference here. Glutamate is an excitatory neurotransmitter, and that's in the entire central nervous system, so brain and spinal cord. GABA, this is inhibitory. This turns off neurons, and it works in the brain almost exclusively. Now glycine, this is also inhibitory, but this is in the spinal cord primarily. So there are kind of two inhibitory neurons in the central nervous system, but really only one main excitatory neurotransmitter. Serotonin, this is an important neurotransmitter for things like arousal, for mood, for pain, and a couple of others. I talked about selective serotonin reuptake inhibitors, SSRIs, and now you know what reuptake is. And SSRIs prevent the reuptake of serotonin. And when you prevent the reuptake of serotonin, you leave more in the synapse. Then I talked about the catecholamines, dopamine, epinephrine, and norepinephrine. Dopamine is really important for things like control of movement, like making movements smooth. Also really important for reward behaviors, addiction behaviors, emotion. Dopamine's a really important neurotransmitter, as we'll see with Parkinson's. Epinephrine, this is the primary fight or flight neurotransmitter / hormone. Really, really important. And then norepinephrine, also important in fight or flight responses, but also really important neurotransmitter in the autonomic nervous system. And I also noted that norepinephrine binds to the same receptors as epinephrine.